We're here today with Dr. Raymond Birge, who's a professor at Rutgers University and is also the co-founder of the biotech startup company Targeron Therapeutics, which we're going to be talking about today. So first, Dr. Birge, why don't you tell us a little bit about your background and how you're interested in cancer research?
Well, thank you, Trevor. It's good to be here. So I did my PhD at the University of Connecticut and left in 1989 to go to Rockefeller University, where I worked on tyrosine kinase oncogenes with suppression. And the work we were doing there was on the mechanisms by which cells become malignant by oncogenic transformation. And there was a great ferment in the early 90s to develop targeted therapeutics and target oncogenes like EGFR, Herceptin, other oncogenes that would drug oncogenic mutations. But unfortunately, what is often observed with targeted therapeutics is the emergence of drug resistance. The oncogenic mutational burden of tumors evolves, and this leads to a problem in maintaining inhibitory pathways. And so I began to get interested more in tumor microenvironments in the late 90s before I came to Rutgers. So,
So what aspects of the tumor microenvironment in particular are you interested in?
Yeah, thank you. I'm interested really in how the cells that are not malignant interact with the malignant cells in order to foster immune suppressing environments. We now know that when tumors, particularly solid tumors, evolve, they've already suppressed host immunity. And this is the reason tumor can progress is that the immune system doesn't see it as far. So we became interested in a particular interplay between the cell death in the tumor microenvironment and macrophages that are clearing apoptotic cells.
And could you elaborate on that a little bit? Maybe there's a specific signal or inhibitory signals.
That's right. So the main form of cell death that occurs is a cell death called apoptosis, which is a very programmed and genetically defined, very evolutionarily constructed. And in our bodies, we have about 1 million cells per second die at apoptosis, which is about 100 million cells a day, 100 billion cells a day. And that's about 0.5 to 1% of all our cells. And when this happens, this should get damaged cells, old cells, to be replaced by new ones. And it's a very homeostatic and necessary part of the homeostasis of tissue for all organs. And so this is not inflammatory. It has to be non-inflammatory. And there are signals set out by the apoptotic cells to keep them suppressive.
And so what is the strategy at Targeron to sort of combat this with your therapeutic?
Yeah, so the signals, going back to the signals in the tumor microenvironment, one of the emblematic signals during apoptosis is the relocalization of lipids from the inside of the membrane to the outside. A particular lipid called phosphatidylserine in homeostatic in normal cells, it's almost exclusively on the inside leaflet of the plasma membrane. Over 98% of the phosphatidylserine is inside the cell. But when cells die, by mechanisms that we are now reasonably well understood, the phosphatidylserine is externalized to the outside. And here it now serves as an emblematic signal for the macrophages to clear it. And when that happens, there's a very strong inhibitory signal in the tissue that this is all okay, this is normal, this is just the way we clear and we reprogram new cells. But in the tumor microenvironment, this process becomes pathophysiological. Tumors are unlike normal tissue because they are in a constant state of proliferation and cell death. Tumors are sometimes called wounds that never heal. And this necessitates the apoptosis and the clearance, what we call efferocytosis, by the macrophages. And they continuously do this, and they produce inhibitory factors that suppress the immune environment, the tumor. And this is a very common feature of malignant solid tumors. Not only is the phosphatidylserine exposed on apoptotic cells, but what we're now finding is that the phosphatidylserine is exposed on the metabolically stressed and hypoxic and the other cells that are going to ER stress and other stress signals in the tumor microenvironment. So now we have stromal cells, we have vascular cells that are also phosphatidylserine is out. And so this creates a vulnerability for the cancer because under normal tissues, phosphatidylserine is hidden away. But in the tumor microenvironment, phosphatidylserine is exposed and available. And the studies that have been done so far, where you can use monoclonal antibodies to target phosphatidylserine or phosphatidylserine receptors, show very promising results to enhance the tumor immunity and have a favorable outcome.
And so what is it about Targeron's platform that allows you to really take advantage of this meeting compared to some other
So the strategy so far have been to use the phosphatidylserine targeting antibodies to mask the phosphatidylserine. And as I said, it has some preclinical value and it has some utility. Targeron's platform is to completely develop a new targeting concept. So Targeron, what we're doing is we're using the phosphatidylserine targeting modality, and we're fusing that to a payload that will enhance the immune response. So when these molecules go and home into the externalized phosphatidylserine, they're bringing in an immunogenic cargo that will re-enlighten the immune system to see tumors. And that is their unique strategy, is to have a chimeric approach. So Targeron is a targeting modality, and it's an immunogenic.
That's really interesting. So are there other areas that Targeron also adjusts, such as cancer?
Yeah. So the strategy that we've used is in collaboration with another co-founder, Dr. Sergei Kotenko, is we're using the payload as a combination of two types of interferons, a type 1 interferon and a type 3 interferon, which was discovered in his laboratory about 20 years ago. And the utility of these two types of interferons is that it's going to engage two types of cells in the tumor microenvironment. It's going to engage an immune cell, which type 1 interferon receptor is mainly expressed on. But more importantly, it's also going to target an epithelial adenocarcinoma, which is where the type 3 interferons are expressed. So the phosphatidylserine is bringing in a dual action interferon, which we think will enhance the immune response in the tumor. Our studies to date show that when we locally produce these molecules, the tumors really don't grow up very well. And this is a very optimistic but an exciting idea that they can enhance the immune response, and particularly T-cell mediated response. What we've shown is that tumor cells, the tumor cells can upregulate MHC class 1 when they bind to this pigment proteins. And that's what you need to get good anti-tumor.
That's great. So you mentioned a little bit about the anti-tumor efficacy that you have. Can you talk maybe a little bit more about the accomplishments of Targeron so far, what you guys have done?
So we've created a series of platform molecules, which as the phosphatidylserine targeting to the interferons. We have a whole pipeline of these molecules, and we've created them, and we've shown they're biologically active in vitro and in vivo, and when they're locally produced, they can have strong anti-tumor activity. So the next steps are to deliver these molecules in a way that they can be reach the tumor and bind and hold it to the phosphatidylserine. That's what our current efforts are on right now, is to target in vivo and to deliver them into the tumors to show efficacy as a therapeutic that is injected into the patient like the mouse.
Okay. Sounds really promising.
Yeah. So another aspect of this that which is important for the interference of what to afford is what are applications, and that are the phosphatidylserine externalization is a very ancient signal. It goes back all the way through evolution, back even to C. elegans and other primitive organisms. And throughout evolution, what we've observed is many, many viruses many envelope viruses will put phosphatidylserine on their surface as a form of apoptotic mimicry. And there's really many viruses. There's SARS-CoV-2, influenza, many viruses, HIV, risk virus, just a huge variety of them. And they all put phosphatidylserine on their surface, and they use this as an immune inhibitory signal. In some cases, they can use it as an entry signal to actually get inside cells. And that concept, when many viruses who are using an evolutionary arms race, really evolving at different evolutionary types, all come to the same consensus to use phosphatidylserine, is very strong evidence. It's called convergent evolution. And when you see that, you see this with P53 too, virus P53, the virus, the endovirus. When you see that, it's very obvious that phosphatidylserine is a very important target that can be exploited. The last thing I'll say about phosphatidylserine is that unlike proteins and glycocalyx, other types of epitopes, which are under tremendous mutagenesis, and proteins mutating, like glycocalyx, dischanging, polysaccharides. Phosphatidylserine is stable as tumor progresses and does not change. The backbone doesn't change. Therefore, it should be a stable epitope as tumors develop, even in solid tumors go into the immune stage. Right.
It's great. Really interesting. Okay, thank you, Dr. Birge.
Thank you, Rick. Pleasure.
Absolutely. Yes. Best of luck.
Thank you.